As recently as the late 1960s, developmental biology was kind of a nerdy field where people who were interested in separating the cells of frog embryos or cutting a frog egg in half and seeing that the frogs could develop, the eggs could develop into whole frogs despite the fact they were cut in half, were similar experiments with sea urchins, an obscure, useless organism. That was what fascinated people. People went into this field, and there weren't that many people in the field for pure scientific interest, kind of like how people went into astronomy and things like that. Then in the 1970s, there was a technical revolution in the ability to manipulate genes and the ability to sequence genes and the ability to take genes and put them from one organism to another. And it kind of reinforced an idea that had been hanging around for a while called genetic determinism, where because if you made changes in genes, you would make changes in the organisms. And some people felt that they had a handle on what made an organism tick. And this drew a lot more interest into this field. I have to say that 30 years on, in the present day, we still don't know why when you separate the cells of a sea urchin embryo, it develops into two sea urchins. We still don't know that, despite the fact that we can pinpoint every gene in the sea urchin or in the human or in the mouse. We don't really know how it works. There's no genetic blueprint that has emerged, and there's no genetic program that has emerged. Okay. Well, having said that, there was a great deal of activity and a kind of a landmark intervention by scientists. Some scientists said, well, if you could put genes from one organism to another or put genes from humans into microbes, maybe you could cause new diseases. And people got concerned, and there was a meeting in Asilomar, California, where a lot of scrutiny was put on this kind of research. And it led to some proposals for restrictions and reform. And it was kind of, it rarely happened in the past that scientists got together and looked at their field and tried to speculate about what could go wrong. And then shortly thereafter, in 1980, a couple of things happened. One was the passage of the Bayh-Dole Act in the US Congress, which was an act that wasn't particularly involved with biology, but said that the government is spending a lot on university research, and it's difficult for people to make money from those discoveries. The discoveries go into the public domain, but people aren't forming businesses about it. So why not allow universities to patent and establish intellectual property in things that were funded by taxpayers? Okay, so that was one change in 1980. A second thing that happened in 1980 was something called the Chakrabarti decision in the Supreme Court. And there, a geneticist that worked for the General Electric Corporation had found a way of getting certain bacteria to eat oil spills. And the General Electric Corporation was in the electrical business, but also partly in the business of making oil spills. And so they were interested in having these bacteria chomp up the oil, and they tried to get a patent for it. And the patent office turned them down, and there were fights through the courts. And finally, it went up to the Supreme Court. The patent office didn't think that living organisms should be patentable, and living organisms weren't patentable before. And through some kind of misconceived notions and some court opinions that said that genetically modified microorganisms are chemical substances, they're not really living things. Even back then, there were no biologists on the Supreme Court, and I don't think there are now. There was the Chakrabarti patent prevailed in the Supreme Court by a 5-4 decision. And then, not too long thereafter, patents were issued not only on microbes, but on mice with human genes in them, and oysters, and human cells, and so on. So kind of opened up intellectual property in anything that was genetically modified. So when that happened, the ability to get patents on living organisms, the ability of people who did research with public money to get patents on their inventions and share it with their universities and their companies. Well, another thing happened in 1980, which was the election of Ronald Reagan. And that had a very interesting effect on developmental biology, because up until 1980 and thereabouts, people who were doing research in this field, who discovered a new gene that might be associated with a disease, the motivation that they would put into their grant applications and into the public relations releases from the universities were, this could be used for prenatal screening, prenatal diagnosis. This became kind of not done under Reagan. You couldn't use that justification anymore. So people started to say, well, we can manipulate genes, and we have this gene that's associated with the disease. Potentially, maybe somewhere down the line, we can make humans that don't have these genes, or that we can correct these genes in human embryos. So there was a kind of a shift in the mentality of justification of biological research. It was no longer the idea that maybe we can use this genetic information to do screening and so on. And there are certainly problems with the eugenics of that strategy. But in fact, it was the abortion issue that made it kind of more respectable to justify this research by prospective genetic engineering. And then the idea of genetic engineering began to take off. In 1988, a person who's sometimes called the father of bioethics, which is a profession that emerged along with the biotechnology enterprise, the University of Virginia professor named Joseph Fletcher, who was also a Protestant minister, in his book, The Ethics of Genetic Control, Ending Reproductive Roulette, called for using genetic manipulation to create parahumans, that is, partially human organisms, or chimeras, that is, mixtures between humans and non-humans that were fashioned to do dangerous and demeaning jobs.